Hi, my name is Adeline Rodriguez, and my learning outcome is 4.6. Describe the steps of protein synthesis. First off, what's protein synthesis? Protein synthesis is the process that cells use to make proteins. This process is efficient, recycling components and translating many copies of a protein at a time. The energy used in protein synthesis is provided by ATP molecules. This process consists of two stages, which are transcription and translation, and which we will talk about in the next slides. The first stage is transcription, which takes place in the nucleus. In this stage, DNA is transferred into RNA. The first step of the transcription stage is initiation. In this step, an RNA polymerase binds to a promoter, which is a DNA base sequence that begins a gene. A section of DNA unwinds and pulls apart, exposing a part of the base sequence. The second step is elongation, where RNA nucleotides complementary to those along the DNA strand are joined, forming a molecule of messenger RNA, also known as mRNA. The last step in this stage is termination. In this step, the RNA polymerase reaches a special DNA sequence or termination signal, indicating the end of the gene. The RNA polymerase releases the newly formed mRNA molecule and leaves the DNA. mRNA passes through a pore in the nuclear envelope, leaving the nucleus. It then enters the cytoplasm, which is where the next stage of protein synthesis occurs. The second stage is translation, where RNA is translated into a protein. At the beginning of a messenger strand, a ribosome binds to the mRNA near the codon. An initial transfer RNA, also known as the tRNA anticodon, recognizes its complementary mRNA codon and brings its amino acid to the ribosome. A second tRNA brings the next amino acid to the ribosome. A peptide bond then forms between the two amino acids, releasing the first tRNA. This process repeats for each codon as the ribosome forms a chain of amino acids. When the final codon on the mRNA is reached, it provides a stop signal, indicating the end of protein synthesis. In turn, a newly synthesized protein is released. In conclusion, in the process of protein synthesis, DNA is transcribed into mRNA, which is then translated into a protein. These are my sources, and thank you for watching.